Kia ora tātou. Thank you for coming out on this beautiful morning to hear me talk, and thanks to Ben for an introduction to the Sustainable Seas program. I won't deal with that at all. In this talk I'll start with a bit of the big picture overview of what phytoplankton is, some discussion about algal blooms in coastal New Zealand, and then get on to the substance of the Innovation Fund project that I've been involved in. So, why are phytoplankton important? Well, all life in the oceans and much of the life on land actually depend upon the invisible plant plankton that's floating there through the window there. They produce about 50% of the oxygen that you're breathing, so every second breath you take, you can thank those little plants out there for that. The world would be a very, very different place if we didn't have phytoplankton in the oceans. This image actually is just a composite image of phytoplankton productivity around the globe, taken obviously through a northern hemisphere summer period, so the colours are more intense in the north. But what you'll notice is that the blue patches in the middle of the oceans are very depauperate areas, low phytoplankton biomass, low nutrients, and most of the productivity is occurring in upwelling areas in equatorial regions like this, or off in the lower, higher latitudes and around New Zealand there. Phytoplankton have an absolutely crucial role to play in essential cycling of elements in the biosphere, particularly with respect to elements such as carbon and nitrogen, of course, of which we're all aware of the importance of carbon. There's incredible diversity of phytoplankton. In this image here, it represents two of the two major groups. The top group here, the diatoms. Diatoms have silicon-impregnated cell walls and elaborate structures to keep them floating. They cannot swim. The images on the bottom are of dinoflagellates, another large group of phytoplankton, very common throughout the oceans of the world. All sorts of types of nutrition, from autotrophic photosynthesis, heterotrophs, mixotrophs doing both. And these organisms can swim, and so there's an essential difference between the water column conditions that stimulate the growth of one or another. There are other major groups. These are just two major groups, but there is another very large group of phytoplankton which inhabit the oceans, and these are very small cells, much less photogenic than these, little anonymous blobs really. They occupy much of the ocean but are incredibly important in the carbon cycle. Things such as chlorococcus and Sinococcus. And there's another group altogether which also inhabits much of the central tropical regions of the world's oceans, and that's the Trichodesmium, a cyanobacteria that fixes huge amounts of nitrogen and is a major contributor to the nitrogen content of the oceans. There's a constant jostling for supremacy in the phytoplankton. It's always changing. There's one species supplanting another. This model here, it's a model based on also on a lot of data. There's a great deal of interest, of course, with respect to climate change as to what is happening in the ocean and how and where carbon dioxide, for instance, is being absorbed and where it's being emitted from the oceans. And this is a model from a project known as the Darwin Project, which has modelled the different sort of trophic levels in the ocean and the activities that are going on. It's a fantastic image to show that variability and the changeability of plankton populations. North and southern hemispheres, the orange colours are largely diatom-based communities, and in the central gyres, a lot of the smaller picoplanktonic forms. So phytoplankton, as I said, very important in the carbon cycle. Here's an example from New Zealand, central New Zealand. You notice Cook Strait there, those milky plumes off the east coast there. Those are blooms of coccolithophorids, these very small cells. The cells have calcium carbonate scales on them, so the water looks very milky, but very important in transporting carbon to the sea floor. There's large areas of the seafloor around New Zealand on the continental shelf that are calcareous sediments that are composed of the remains of these cells. Often what happens on land, as Ben points out, affects the coastal waters. This is an example of a very intense bloom of a dinoflagellate off Napier a couple of years ago, after a period when they'd had very heavy rains in the catchment of the area. These things are quite common. Another example actually off out in Canterbury, off the Ashburton River, a major bloom of another dinoflagellate altogether, extending way along the coast there. Noctiluca scintillans, something that you probably see around here a bit, responsible for the very bright bioluminescence you sometimes get in the seas during summer. Many of the dinoflagellates actually do bioluminesce, but Noctiluca is certainly one of the brightest. We get lots of reports of Noctiluca blooms around the Sounds and other parts of New Zealand. It's not a photosynthetic dinoflagellate, it actually grazes on diatoms, this particular species. And in the Marlborough Sounds, which is the main area that we're interested in, as Ben pointed out, because of the presence of the aquaculture industry and its economic importance to the region and the nation. We're often tracking blooms of things. This is actually a non-toxic bloom very recently, but we're making more and more use of satellite images where weather permits. But in this particular image, you can see in these enclosed bays, there's a bloom going on there, a bloom going on there, another bloom going on there. They're all the same species. This, yep, so this picture here is how dense it can get in the inner waters of town. Are they? It's not changing. I see it goes. A particular favourite of mine, this goes back many years, it's really the reason why I got involved in this area, was a bloom back in the early or mid-1980s, known as the Tasman slime bloom, and it was a very mysterious event. The bloom basically caused the water column within the bay to turn to jelly, really, and at the time the snapper fishery had been overexploited and snapper had pretty well gone. The scallops had been dredged to bilio and there was practically no scallops left, and all they had left was fishing for sharks, gill netting for sharks and so on. And then this bloom came along, they couldn't even do that, so there was quite a kerfuffle at that time. And it was very mysterious, but eventually we solved the problem, and it turned out to be a dinoflagellate that produced really copious quantities of mucilage in the water. And the most satisfying thing about it was this was a conundrum that was many years old, for millennia. It had been noticed in the Mediterranean Sea, in the Adriatic. This phenomenon had been known as Mare Sporco, or Dirty Sea. And as a result of this bloom in Tasman Bay, we were able to determine what the cause was. Anyway, the real reason why we get paid to be interested in this thing is because the potential effect of algal blooms on the aquaculture industry and on contamination of shellfish, and it's important for the health of consumers of shellfish. And some algae, microalgae, and they're in the minority, I would say, there's only a relatively handful, but produce toxins that accumulate in shellfish, and when people eat them they can get sick, and some of them are really potent. There is one in particular known as saxitoxins, which are highly potent neurotoxins, and there's very strict international regulations about how much you're allowed to have in shellfish. And woe betide the mussel industry, where they export any product overseas and over the limit, it would be extremely serious for the future of the industry. So a lot of our time is spent trying to advise the industry on how to avoid these problems. And an early warning of the blooms is one of the main things that we're capable of doing. Oh, I should have mentioned too, it's not only the impact on shellfish, but also there are a number of species that are highly toxic to fish in sea cages, and the salmon industry is a fairly important industry in the Marlborough Sounds, and they from time to time have problems of this nature. So the situation's actually changed recently in the Marlborough Sounds. It's actually become somewhat worse, and we're not quite sure why. It's certainly coincidental with large-scale changes in water temperature in the region and in the Tasman Sea, the marine heat waves that have been going on for the last couple of summers. And a lot of it's been caused by the sudden appearance of a toxic dinoflagellate known as Alexandrium pacificum, first observed in Queen Charlotte Sound in 2011, at which stage it closed the shell fisheries in Queen Charlotte Sound for some months. It's bloomed every year since then, and we were on tenterhooks for several years wondering, is it going to actually penetrate into Pelorus Sound, which is the real centre of the mussel industry. And then last year, in May last year, it suddenly appeared, and it appeared in a big way in the central part of the Pelorus Sound, and with causing red tides, highly discoloured water, very high cell concentrations. The bloom appeared to originate anyway, in a bay in the central part of Pelorus Sound here, indicated there, and then spread from there and contaminated shellfish throughout the whole rest of the sound. There was about 250 mussel farms were closed for a period of up to 3 months, and caused major problems, of course, for the industry. And then it has appeared again this year. It appeared somewhat earlier, but bloomed most intensely over the winter months. And the harvest closures extended through until near the end of July. The figure on your right there is just shows the levels of toxicity. The red is where the levels are actually over the regulatory level of 0.8 milligrams per kilogram of flesh. But and although it was relatively, these high levels are relatively kind to the central part of the sound, other areas were extended lower, but the whole area had to be closed because they couldn't be certain that they would get caught out. The industry has become very gun-shy about this, and any hint of it affecting their mussels, they close down until they're absolutely certain that it's clear. There are a number of areas where we know that the blooms originate. One is in this bay, in Nydia Bay in Pelorus Sound. There's another inlet in Queen Charlotte Sound, the Onapua Inlet, and another area in Port Underwood. All these are mussel growing areas around here. And you'll notice one thing about them, that they're all long enclosed inlets with limited water circulation. And quite a bit of our work is to try and determine what it is that leads to these blooms occurring. And in many cases, it's relatively obvious. In this case, this is the Onapua Inlet off Tory Channel. If you've been on the ferry from Wellington to Picton, you'll go through Tory Channel here, and as you before you come into the main part of Queen Charlotte Sound, you'll pass this on the way in. And it's quite a long enclosed inlet. During summer, the water temperatures increase in this area, and water column stratification develops. There's an interesting feature of Tory Channel in that it's a very turbulent area. It always has high nutrient concentrations because of the continuous upwelling that's taking place from deep water from Cook Strait, ebbing and flowing through the channel. So the high nitrate levels influence the inlet. During summer, water column stratification develops ideal conditions for the growth of dinoflagellates, of which Alexandrium pacificum has become a major component, and that's essentially the scenario that takes place. An important feature of Alexandrium pacificum is the fact that it has a benthic stage to its life cycle. And as a result of these blooms, there has been the development of quite high densities of cysts in the sediments in these inlets, and every year this is where the blooms originate from. So the cysts hatch. The cysts are actually the diploid form of the stage of the life cycle. The vegetative form is the haploid stage, so it divides by binary fission in the water column. Given some environmental trigger, of which we're not totally sure what it is, it doesn't seem to be nutrient limitation, which is the general dogma. It more likely seems to be competitive interactions with other components of the phytoplankton. It then will suddenly disappears from the water column. Large numbers of cysts appear in the sediments. And in these inlets, like our Onapua Inlet, we have high numbers of cysts in the top end of the bay, and this is where every year the blooms are originating. This was a image from July. It's a very fortunate event, really, and we were able to really see for the first time, because it just so happened that there was no cloud around for a few weeks, the development of the bloom up here in the top part of the inlet, where the cysts were actually hatching from the sediment. The significant thing to notice about this image is the yellow dots there are actually the routine monitoring stations where every week samples of water and shellfish are taken and analysed for the presence of toxins and for the presence of toxic dinoflagellates. And you'll notice that although it was only a matter of a few hundred metres, maybe half a kilometre from this innermost site, the conditions were completely different at the site. And so this is one of the issues, that the patchiness of these types of blooms is a problem for the monitoring program. Just to point out, along this coast here, you'll notice there's these shadows. That's a continuous line of mussel farms along that area. This is actually one of the areas where mussel farms were first developed in the Marlborough Sounds. So it had no problems for over 30 years. Suddenly it's now got a major problem. And they haven't actually been able to harvest in here really for nearly a year. There's quite a good relationship between the numbers of cells represented by the orange line there and the actual toxicity in the shellfish. The red line there on the bottom is actually the regulatory level of 0.8 milligrams per kilogram, and harvesting shellfish over that limit is not permitted. Monitoring of the cells in the water column does give it quite a good early warning if the samples are collected at the right frequency and in the right place. And it's a very laborious process. There's a whole team of people at Cawthorne Institute who spend all their time looking down microscopes, and there's a sort of rogues gallery of cell types they're looking for. Very tedious and time-consuming thing, but it works. But the Innovation Fund project that we've been working on is to try and improve the situation by developing some rapid tests that we can do more samples more frequently for lesser cost, really. So the first method we were trying was a portable DNA tracking system using a technology known as hydrogel quantitative polymerase chain reaction. And this was essentially a kit produced by a Canadian company. It was originally developed for diagnosis of tropical diseases, whereby all the reagents for the reaction are contained in a gel, and all you need to do is add a crude DNA extract to the tubes and run it through the thermocycler, which is this little instrument here. And it works okay, but not perfectly. What it has allowed us to do is you can take the assay on a small boat, and you can run within an hour and a half about 16 samples. The DNA extraction and everything is relatively simple, and it's certainly very competitive in terms of the time it takes, the labour involved, and the cost. However, we've found that there are problems with the consistency of the gel reaction tubes, and although the sensitivity is fantastic, you can detect cells at levels way lower than you can ever detect by microscopy. The precision of quantification is really not up at this stage. Is really not up to being reduced as a regulatory method. The second type of technology that we've been trialling is using this instrument here in this photograph. It's known as an imaging flow cytobot, and we have had for the last 6 months or so one of these instruments on loan from a colleague in the University of California. And what it is, it's a flow cytometer that takes pictures of all the particles as they flow past the sensor. And it's been specifically designed for the analysis of phytoplankton, developed at Woods Hole Oceanographic Institute in the United States, and now manufactured by this company called McLean Lab. As I've said, we've had one on loan, and we've been trialling it. What we would hope to do with this instrument is to have it actually on the sampling vessel, so that as one is moving along through the sounds, you can take water samples, put them through the instrument, and get within 15 to 20 minutes have a characterisation of all the cells that are in that water sample. Use of a microscope normally on boats doesn't work. It's just too difficult. But through these means you can do it. So, and this instrument, not only does it count the particles, it sizes them, and it has got imaging analysis software and cell classifiers that are being developed to actually recognise and identify the cells as they come through. So we've tried it by travelling around the sounds, analysing samples, and it works really well. It can be remotely deployed In this case, the image on the right there is it on a salmon farm out in Pelorus Sound, where we had it for 3 months, every 20 minutes sampling the water column and telemetering the data to us back at home. And quite a surreal sort of situation really, to be sitting at your desk 100 kilometres or so away from where this is all happening, and being able to see the sort of the intimate goings on of the plankton as it happens pretty well. Quite a revolutionary situation actually. This is the sort of raw data you get from the instrument, loads and loads of pictures of cells, and the ones that we're really concerned about are really relatively easy to spot. These ones here, this one here, this one here, amongst all the others. But what it does tell you, it gives you everything in the plankton. And as I sort of mentioned before, it seems to be the interactions between different elements of the plankton that really are determining the longevity of the bloom, their intensity, and the duration. And so this is work still in progress. There's good comparison between the sort of conventional microscopy estimates and estimates we get from the instrument. So in summary, these new monitoring technologies can enable higher phytoplankton sample frequency and greater spatial coverage, which is really what we need to provide greater security to the industry. They can provide early warning of blooms developing, so you're detecting the cells at low numbers before they've developed sufficiently to push the levels of toxicity over the limit. Protect the public and importantly protect the industry and ensure market access for agriculture products all around the world. These molecular methods we've been trying, they certainly are promising, but they at this stage need further development to reach the level of precision that's required. The imaging flow cytobot works really well, and it could be introduced now to supplement the current monitoring program, and we're trying to convince the industry that this is probably the way to go. And a number of people have helped with this study, and thanks to them. And that's it. Thank you.